Thanks for the stage, Adriano, and the introduction. I think first thing I'd like to say is congrats to Jim and thanks to Jim. A lot of the data that I'll display today was developed by Jim's team. And so I'm not surprised to see him get such an amazing award. Thank you again, Jim, for all the support in the past. So I'd also say that I'm going to sort of give this narrative style presentation, but I'd like to make sure everybody in the audience knows that that's not always the best way to interact with us. The best way is after you see something tantalizingly interesting in one of these presentations, reach out to us, reach out to Adriano, get us on the phone, get us on a video call, and then we can talk very specifically about all of your needs and use cases. That's when the rubber hits the road, and that's when we all start to have a lot of fun talking about science. Today, the title is Testing the Efficacy of Immunomodulatory Antibodies in Humanized Mice, and I'm going to jump right in. At JAX, when we think about the humanized immune system mice, the first thing that comes to mind is the model that we tend to work with, which is the immunocompromised NSG mouse. The NSG is on the NOD or non-obese diabetic background, and that means we've got an absence of hemolytic complement, reduced function of dendritic cells and macrophages, and optimal HSC engraftment environment due to the SERP alpha allele mutation, meaning that these MACs and dendritic cells do not recognize these human HSCs as non-self. They also contain the SCID or PRKDC DNA repair mutation, meaning that you don't get VDJ recombination in T and B cells, and the IL-2 receptor gamma mutation, meaning that you don't get mouse natural killer cells. And what we do with this mouse is we humanize them, and we tend to do it two different ways, either through CD34 positive hematopoietic stem cell humanization or through PBMC humanization. And when we're doing CD34 positive humanization, we're irradiating the mice, and then we're injecting these human CD34 positive hematopoietic stem cells. We're waiting approximately 8 to 12 weeks, and now you've got a humanized mouse. They come in three main varieties that we tend to work with, although we're always expanding these. The three main varieties are the baseline CD34 humanized NSG mouse. It's the gold standard right now. Got strong T cell representation and a little bit weaker myeloid lineage and natural killer cell representation. And that takes us to the CD34 humanized NSG SGM3. This SGM3 transgene contains IL-3, GM-CSF, and stem cell factor. It was originally developed to promote and improve AML engraftment, but what we found is it shows very strong T cell levels as well as improved myeloid cell development, and still in this case few natural killer cells, leading us to our CD34 humanized NSG IL-15. This one supports functional human T cells, a lower level of myeloid cells, and most importantly, physiological levels of natural killer cells, if that's a population you're interested in working in. The second variety of humanization that we tend to practice on a regular basis is direct injection of PBMCs. And we do that for two purposes typically. When we're working in the PBMC humanized NSG, we know that this is going to be a short-term human immune system model, because what's going to happen is you're going to have development of acute xenogenic GVHD. And that's great if that's what you want, right? So that's what we use this model for, is for the development of that GVHD and to allow you to use your immunomodulators to treat or prevent that GVHD. GVHD development tends to take around 2 to 6 weeks post PBMC engraftment. But what if you want to do some immuno-oncology work? Well, we also have the PBMC humanized NSG MHC1 and 2 double knockout. The major histocompatibility complexes in the mouse tend to be the primary mechanism of this acute xenogenic GVHD. There's still a non-canonical route by which GVHD occurs, but you can see it takes a whole lot longer for that onset, giving you a nice experimental window for your immuno-oncology experiments. And I'll show you some diagrammatic layouts of how that might work. Here, I like to give you a chart that gives you a nice overlay of what's going on. This column right here is the mouse type, and then each of these subsequent columns are representations of various T cell levels, myeloid cell levels, natural killer cell levels, or GVHD that you might expect to show up in the model. And here is where the T cell selection occurred. And that's an important thing to understand. And let's first talk about the CD34 varieties here, the NSG, the SGM3, and the IL-15. We know the NSG has strong T cells, weaker myeloid natural killer cells, and essentially no GVHD. We know the SGM3 has stronger T cells, even driven by this transgene here, stronger myeloid cells, similarly low natural killer cells. And I've got a little asterisk here just to remind folks that occasionally we do see macrophage-induced anemia or mass in a very small percentage of donors in the SGM3. And if you're curious about that, please feel free to reach out to us afterwards and we can discuss. The IL-15 mouse has strong T cells, weaker myeloid, and that physiological level of natural killer cells. In all three cases, the T cells have been selected for in the context of the mouse. And so that means that the affinity and the avidity of those T cell receptors bound to their target antigen was selected for as displayed in mouse major histocompatibility complexes. So that may or may not be problematic for your research, so it's something for you to think about. That's one advantage of working in the PBMC models, where you'll have the T cells having been selected in the human thymus before being drawn from a human's blood. And you'll remember the PBMC humanized NSG has a strong T cell engraftment. The myeloid and natural killer cells tend to fall off rather rapidly, and giving you the GVHD model that you can then try and suppress with your therapeutic. The PBMC humanized NSG double knockout, on the other hand, has your strong T cell representation, as well as a vastly decreased GVHD so that you have a nice experimental window. Now, before I jump into the rest of the talk, I just wanted you guys to know I was introduced as a study director. And what that means is I work as part of a commercial group in our Sacramento facility, running commercial studies as a CRO style function for people like you. So if you find yourself in need of some in vivo oncology experimentation, we support syngeneic studies, even in aged mice. We do spontaneous and induced mouse cancer models. Today, the ones circled in red are the ones we'll focus a little more on today, but we work in human cell lines, human solid tumor PDXs, AML PDXs, and CD34 and PBMC humanized immuno-oncology, which is really the focus for today. As well as acute xenogenic GVHD. But I didn't want to leave out the fact that we run these in vivo cytokine release assays. I know Jim will speak more about this. This is one of the many things Jim's team invented and brought to the fore. It's a very exciting tool. So please don't hesitate to come out and ask us about it. We are also doing CAR T work for many groups, and some of them are asking us now about safety studies. We've run many safety studies in the context of CRISPR modified hematopoietic cell therapeutics, and we run many engraftment assays looking at people's HSCs. So let's talk a little bit about PBMC humanization for acute GVHD. I'm going to go over experimental design, donor selection, and show you some data so you understand what the disease model actually looks like on the ground. The experiment that we would lay out typically is going to be run in the NSG mouse. Usually folks will only look at one donor at a time, but you do have the option of selecting multiple PBMC donors. We irradiate the mice, and then we transfer 10 million PBMCs through an IV injection. Dosing can either be prophylactic or it can occur after we've done irradiation and PBMC injections. The controls we tend to work with are things like belatacept, abatacept, and Enbrel. And the readouts are typically body weight, disease activity index, and flow cytometry at various time points or single time points. And this could be the simplest study design that you might see. Before we get started on a study for GVHD, what we'll do is we'll share with you some of the data sets that we have to allow you to make a rational decision about what you expect your disease onset to look like. Here, what we're looking at is 4 different donors denoted by blue, red, green, and pink on a Kaplan-Meier survival curve. And you can see that blue has a relatively slow onset, going almost all the way out to, let's say 55 days here before all mice succumbing to GVHD with no therapeutic intervention. While the other 3 donors are all rather aggressive, right? We can see onset probably somewhere around 12 to 13 days here for this green one. And we allow you to choose because we know you, in consultation with us, will be able to evaluate your MOA and figure out what would be the most effective and most reasonable route to take for testing your drug in this model. Here is an example study, and this study was actually run by us looking at abatacept, belatacept, and vehicle. Everything here is the same with the exception that we're specifying that we're doing prophylactic dosing starting at day -1. And we are going to use the multi-parameter FACS immunophenotyping to get an idea of what's going on. The expectation is that abatacept and belatacept will be able to block CD80-86 from co-stimulating the T cell and therefore block inflammation. So what you're looking at here is percent body weight change on the top right and the Kaplan-Meier survival curve. I do apologize because there's an extra test article group here, but we're going to focus in on the orange, green, and black of abatacept, belatacept, and vehicle. And when you look at orange and green here, you can see that the abatacept and belatacept showed nice increases in percent body weight throughout the study, at least out to about study day 32, whereas the vehicle starts to show a drop off of body weight. And this is the end point for a big chunk of that group before things start getting a little bit noisy here. And when you look at survival, you can see a similar pattern, mice beginning to come down from the vehicle group. And we did actually lose a few mice from the belatacept group. And we'll be able to tell what was going on there from the flow cytometry data. So I'll jump to that next slide. This looks a little busy at first, so I'm going to go ahead and outline what the different quadrants are. The top 2 graphs here are both focused on CD4 positive helper T cells at time point day 10 or at time point day 35. Day 10 should be pre-phenotypic GVHD onset. Day 35 should be a whole bunch of mice have come down from the study if they're going to. The bottom 2 graphs are focused on CD8 positive cytotoxic T cells at study day 10 and at study day 35. And then on each one of the graphs, we're looking at total CD45, CD4, naive, effector, central memory, effector memory, CD69 is an activation marker, and Treg markers. And the real interesting ones that pop out here at study day 10 is that early on with abatacept and belatacept, you're already blocking some degree of T cell proliferation. And we know that the majority of the proliferation in a model like this is going to be ultimately leading to disease. You can also see a similar pattern in helper T cells. And you can see that there's a strong level of naive T cells in the context of both abatacept and belatacept, whereas in the vehicle group, these T cells are becoming activated, and you can see them already in the effector memory population right here. And when we take our helpers and we jump over to study day 35 to see what's happened throughout the study, what we see is the CD45 levels in the vehicle control have jumped significantly at this point. And to some degree, so have the belatacept, but the abatacept has stayed quite low. And when we look at naive, we see the control has no naive left, the belatacept has no naive left, and the abatacept has successfully maintained a bunch of naive immune cells. And again, when we jump to the effector memory cells, we see the opposite pattern where abatacept has very few effector memory while the control and the belatacept have jumped up pretty significantly. And when we take that and we look at the cytotoxic panels here, you can see a similar pattern in total CD45, of course, but again, also in total CD8s, a similar pattern where abatacept was very effective at keeping the cell population from proliferating as well as becoming activated and producing effector T cells. You can also take a look at the total CD4s versus a total CD8s. And this is always interesting. And those of you who study infectious disease know that when you have a ratio of CD4s to CD8s that's somewhere above twice as many CD4s as CD8s, you're probably looking at a healthy individual. And then as soon as you come over to the other side on study day 35, and you see that in the control, you have maybe 3 times as many CD8s to CD4, you probably have an infection. Here you have 2 times as many CD8s to CD4s. And in the successful abatacept group, you still have double CD4s of your CD8s. So now let's jump to immuno-oncology using the CD34 and the PBMC humanized models that I described before. I'm going to give you an overview, talk a little bit about timelines of the total experiment, and I'm going to do some comparisons of the models, looking at critical variables, experimental designs. We'll go into the selection of PBMC donors. It's a bit different from the selection of PBMC donors in the GVHD model. We'll talk a little bit about test articles and mechanisms of action and how you might think about using those test articles in each one of these models. And I'll show you some nice data that came from Jim's product development team, really showing how effective these models are, especially in the context of bispecific molecules. So this is a diagram intended to help walk you through the differences between an immuno-oncology model and a CD34 humanized mouse versus a PBMC humanized mouse. So let's talk about CD34. In the case of CD34, we're taking the NSG or the NSG or the SGM3 or the NSG-IL15, and we're going to inject some CD34 positive cord blood cells, IV, post-irradiation, and we're going to wait. And what we're waiting for is for the human immune cell levels to come up in the peripheral blood of the mice, and we'll test that. And once they've passed our tests, we've got a CD34 humanized mouse. These mice can then be implanted with patient-derived xenograft tumors or cell lines, or experimented without in different immunological settings. And now you have your mouse and you're ready to go. The PBMC model is a little bit different, and I'll explain to you why in a couple of future slides. But in essence, it typically makes sense to engraft the PDX or the CDX first in your NSG or NSG double knockout. We would highly recommend the double knockout just because you worry so much less about the GVHD onset. There will be variable timing to the growth and establishment of that tumor. And once you've got that tumor, then you would PBMC inject. And so you'd have your humanized, PBMC humanized tumor-bearing mouse ready for your experimentation. Now, what does that look like in timelines? When we're working with the CD34s, you've got that 8 to 12 weeks up front to establish your humanized mice, and then you can go on to growing your palpable tumor and beginning your dosing study. That might take about 4 to 8 weeks. And 2 to 12 weeks here is intended to give you an idea of what cell lines versus PDXs might look like for a total growth period. When we're working in PBMCs, we implant the tumor first, so that's that 2 to 12 weeks right here. That's the first step we take. Then we PBMC humanize, and that really takes no time at all, followed by moving into your dosing study. So you can see here, this can only be, approximately 6 to 20 weeks, depending on how aggressive the tumor model is, whereas this one can take 14 to 32 weeks. But one wonderful thing that we at the Jackson Laboratory have done for you is make these CD34 humanized models readily available off the shelf, meaning that this wait time, this isn't something that you need to use to differentiate the use of the CD34 versus the PBMC, because we've eliminated it. These mice are on the shelf, ready for you to purchase. You can just call our customer service, honestly, and they'll send you a cohort. So don't let that be the factor that makes you choose one over the other. And instead, let's take a look at some of the other differentiating factors. So let's just compare them a little bit. For the and here I've got a column of CD34 and a column of PBMC. The CD34 humanization is relatively stable, meaning that the changes in the percentages of cells present in the blood, those tend to happen over weeks or even months, and they tend to plateau for a while. Whereas in the PBMC humanized, you tend to see a rise and then a peak and a fall, and you really need to be a lot more aware of what these kinetics look like and prepare for them in the context of your study. In the context of CD34 humanization, GVHD really isn't a consideration. These mice don't really develop GVHD, whereas in the context of PBMC, even working with the NSG MHC1 and 2 double knockout, that will be the upper limit on your experimentation timeline. So you've got to think a lot about the right tumor models to pick and make sure that their growth patterns fit within that window of experimentation that you can use. The CD34 models, as you saw before, can support T cells, myeloid cells, and natural killer cells. The PBMC model really only has T cells, as the myeloid natural killer cells, they tend to drop off about 8 to 14 days post-engraftment. I've seen people try and do an end run around this where they do things like subcutaneously inject the PBMCs with maybe antigen in order to try and get a pseudo-style lymph node before those other cells begin to disappear. And if that's something you're interested in, again, like I said at the top, reach out to us. Let's have an interesting conversation and see what we can do for your particular needs. In the context of the CD34 humanized mice T cells, they're likely showing a primarily allogenic response due to the selection of the T cells in the mouse thymus. Whereas with the PBMCs that have been trained in the context of a human thymus, they're more likely to show an antigen-specific response. So let's say your therapeutic, upregulates tumor-specific antigens. This might be a more useful model in that context, although I do always suggest that we run small pilots across a couple different models to find the one that really does work best for you. Donors cannot typically be reused in the context of CD34, but we are trying. And if you're interested in that, please reach out and let us know. Whereas with the PBMCs, donors can be reused, although there is some degree of variability in oncology experiments, we do see that the engraftment absent a tumor is quite repeatable. Similar and how they're not. And I'm just going to go through how they're similar first. And I'm going to say, in both cases, you're picking a donor or multiple donors. And that donor represents a genetically distinct human being. In the case of CD34, we often suggest that you choose multiple donors so that you're looking at the genetics of multiple human beings. And that's based on our experience, at least with checkpoint inhibitors, that not every human being responds to pembrolizumab. In fact, it's closer to 30% of donors. That's really something to keep in mind. PBMC donors, what I'll often see there is that folks will like to screen maybe like 5 or 6 donors to see what the responses look like in a very small pilot experiment, and then select a few of those donors to expand to efficacy. Appropriate controls. In both cases, you're really going to want to think a lot about what your appropriate controls are. We differentiate controls into things like gold standards because we don't want people to think that things like checkpoint inhibitors are ultimately controls. They really are gold standards. Enrollment tumor volume. In both cases, you're going to most likely have an enrollment tumor volume, although that sometimes changes in PBMCs. And it's something to think about for any experiment that you're designing. And powering your experiment. Again, why we love to lean on pilot studies because it can really help power your experiment appropriately. Then timing of your therapeutic intervention. So remember, with CD34s, we have our immune system in place, we implant our tumor, and we wait to a defined period, whether it's a defined period of time or a defined size of tumor, and then we have intervention. With PBMCs, we inject the tumor, and then we have a defined period at which we inject our PBMCs, and then we'll have a defined period for the therapeutic intervention. Where these things tend to differ is largely about the selection of the cancer model. CD34 easily supports both cell lines and even slow-growing PDX models. PBMC, because of that window that you can work in, we really don't recommend that you work in anything other than the fastest growing PDX models and the most reliably growing PDX models. Either that or cell lines work quite well in the context of PBMCs. The growth rate of that tumor model is the critical factor for your selection, and the timing of PBMC injection is something that's wholly different than the CD34 model. And let's take a look at how some of these might be designed. It's fairly straightforward, but when we're designing a CD34 experiment, we'd be looking at the NSG, the SGM3 or the IL-15, depending on your mechanism of action and what sort of cells you'd like to have present in your experiment. You'd be selecting a cell line or a PDX. Donors, we'd typically recommend 3 or more, and we'd be looking at a tumor volume range for enrollment. So here you can see the 15 or 5 mice per donor reflected in each of these spots. Oftentimes folks want their control along with a combo with their test article and the control. For PBMCs, the consideration is usually a cell line, 1 donor or more, but more typically 1 donor, a tumor volume range for enrollment criteria, and we've got to think about that timing of PBMCs versus tumor injections versus therapeutics. I have had people work with slow growing tumors and just they timed their PBMC injection a lot later and then had their therapeutic intervention almost immediately after the PBMCs. So how do you select these PBMCs? This is something else that Jim's team really did a ton of work to help us understand. And what they did was they collected survival data. And I don't expect you guys to be able to read everything on here. I'm going to walk you through what this stuff is. And I apologize for the size of the font. They collected survival curves on various donors so you could understand what this experimental window looked like for these PBMCs. They looked at body weight change as another stand-in of how healthy these mice are over time. You can see these look really good. And keep in mind, you can't just buy these MHC double knockout mice and throw PBMCs in there and expect to see these exact same results. Jim's team spent a lot of time identifying the best PBMC donors for this kind of work. And here you can see we've got CD45, CD4, and CD8. And when I said these engraftments were kinetic, this is over 70 days. So you see what I mean now, right? T cells or total immune cells are coming up and coming down at various time points. Same for the CD4s and for the CD8s. So those are important considerations as we go into planning these kinds of experiments. Here is an example of the kind of data set that you'll have available to you when you're selecting your PBMC donors to run a PBMC immuno-oncology study with us. Again, I don't expect you to read everything here, just to know that you can get things like the HLAs if that is of interest to you for HLA matching for your experimentation. So let's talk a little bit about specific test articles. This tends to be the way the conversation shapes when we're trying to figure out the best ways to use these models for our clients. If someone brings us a checkpoint inhibitor, the first thing we'll say is keep in mind when we run pembrolizumab in the context of CD34 humanized mice, we do see that about 30% of donors respond. In PBMCs, it's a little bit trickier. The checkpoint inhibitors show a variable response rate, and we have seen some variation even in repeated studies with the same PBMC donors. So I think that choosing your best model for a checkpoint inhibitor, again, a good pilot study will help you differentiate, but I always tend to lean towards the CD34 because it's become such a clear response for us in these mice. Bispecifics are a great story. I'm going to show you some data from Jim's team again on this. And in essence, what they showed using an EGFR CD3 BiTE was that you could start with relatively small tumors or large tumors using the MDA MB231 model in the context of a CD34 humanized mouse and show fantastic results, and that the exact same thing exists for the PBMC model. So you don't need to feel limited for bispecifics to one or the other. They both will work quite well. For CAR T cells, and I know we specified antibodies, but I figured I'd include some thoughts around CAR T cells. For CAR T cells, a lot of folks have been coming to us and asking us about how, especially in the context of the allogenic off-the-shelf CAR T cells, they've been asking us about using the CD34 humanized mice to test for host versus graft, as well as efficacy studies in a more whole human immune system environment. And in other cases for graft versus host as well. So for CAR T, in the autologous context, we've had groups come to us and ask for a true autologous experiment. And in this case, what they wanted to do was take a subset of CD34 humanized mice from the total cohort, euthanize them, collect the T cells, transduce them to generate the CARs, and then inject them into other tumor-bearing mice from the same genetic cohort in order to produce this sort of true autologous model. So that's another option open to you working with the CD34 humanized mice. This is that promised data from Jim's team. And what we're looking at here is the EGFR CD3 BiTE in the CD34 positive humanized NSG. And you can see how big these tumor volumes were at enrollment. And despite that, a very successful intervention with the bispecific antibody. And here are the individual curves down here. A couple of guys that grew a little bit slower, but absolutely beautiful response from the therapeutic treatment. In this next slide, we're looking at the PBMC version of the experiment. So here we have PBMC humanized mice. And again, this is MDA-MB231 with the EGFR CD3 BiTE. In this case, we're displaying both the normal, we might call it normal enrollment range, the lower volume, and the higher enrollment range. And again, the controls go up through the roof and the treated groups come down quite beautifully. It's textbook. So towards the end of the talk, I wanted to come back around to the slide that Adriano showed you and let you know that we are here through a CRO function. My team out in Sacramento, we would love to meet you. We'd love to help you with your hit to lead, with your lead candidate preclinical validation in the context of our PBMC humanized strains and our CD34 humanized strains. Please feel free to get on the horn with us and have a discussion because we know that in that context, we'll be able to help you find your scientific goal, even if I didn't cover it in the talk today. And with that, I'll say thank you for your time and your attention. Thanks to our product development team, our technical information scientist team, our sales team, and our marketing team for setting this up. And thanks to the mice, of course.